So I know what you're saying to yourself. You're saying, Mr. Razor, I appreciate that summary of transcription and translation, but I'm the kind of biology student who really wants details. I need more vocabulary terms. I need more specific and complex information to stuff into my brain to get ready for this final exam. Well, because I get so many emails like that, we're going to go ahead and add a few more details to our information here in Chapter 17. This is Concept 17.2, all about transcription. And it says transcription is the DNA-directed synthesis of RNA, and we're going to take a closer look. So right now we're going to focus on this area right here inside the nucleus of a eukaryotic cell where transcription takes place. This is the first stage or the first step of gene expression. The DNA is unzipped or unwound. One of the DNA strands is used as a template to create messenger RNA, and from there we leave the nucleus, and we'll talk about that in just a moment because that gets into translation. So for the moment, transcription inside the nucleus of the eukaryotic cell. So let's get down to the nitty-gritty of the molecular components of transcription. The RNA strand, the single-strand RNA that we're going to be taking a look at, is created or synthesized by something called RNA polymerase. RNA polymerase, of course, is another enzyme. It's what pries the DNA strands apart, and it hooks on to one of the DNA strands, the template strands, and then it brings the various RNA nucleotides together to form a chain of nucleic acid called RNA. The RNA strand is going to be complementary to the DNA template strand. In other words, much like the DNA where we have the base pairs that match up, A with T and C with G, we're going to have the same base pair matching, complementary matching going on with RNA synthesis. The only difference is with RNA, remember, it has uracil instead of thymine. So instead of A matching with T, A would match with U in this case. The RNA polymerase works only in the 5' to 3' direction, similar to what we've discussed previously. But the difference here is that the RNA polymerase can start a new RNA chain from scratch. Unlike the DNA enzymes that we discussed in the previous chapter who needed an RNA primer to get it started, the RNA polymerase can go ahead and start a brand new RNA chain without any such primer. The area or region of the DNA sequence where the RNA polymerase is going to attach is called the promoter, and that promoter is going to be a little bit downstream from the actual area that's going to be transcribed. So down here in this picture, see this area of the DNA strand right here is called the promoter, and then the area beyond that is actually the area called the transcription unit where it's going to be read and made into the messenger RNA. In bacteria, the end of the sequence, there's a spot called a terminator, and it's a specific sequence of nucleic acids, nucleotides. In eukaryotes, it's a little bit more complex. Some more stuff has to happen after the termination sequence, but we'll talk about that in just a moment. The piece of DNA that we're going to be making a copy for RNA from the template is called the transcription unit. So the section of DNA, like from here to here, that's the transcription unit. That's what the RNA polymerase is going to use as a template to create the messenger RNA. All of this first portion of transcription is called initiation. So the latching on of the RNA polymerase at the promoter site, the unzipping of the DNA, and then the actual transcription process moving down into the termination signal. All of this is considered, or the initial latching portion is considered initiation. Here's a great little quick animation, of course. You can right-click and hit play. Always good to watch these animations because it gives you a more thorough understanding than more than words alone can do. We also have some illustrations we'll take a look at right here. So here is the part we call initiation. Initiation is where the RNA polymerase latches onto the promoter area of the DNA, and it will unwind the DNA at this portion, and then it will move down using one particular strand of the DNA as the template, and that area is called the transcription unit, and then it will reach a termination point where in bacteria the RNA releases, and then you have a strand of messenger RNA, and you're good to go. In eukaryotes, there's a little bit more involved, and we'll talk about that. So we latch on here, the RNA polymerase latches on right here and begins to move down the strand. In this case, the lower strand of the DNA is going to be our template, and the upper strand is not. In some cases, it's switched. It depends on the gene. Any particular gene will always use the same strand of DNA, but not every gene uses the same strand. So some genes use, in this case, the lower strand. Some genes may use the upper strand, but they are specific to the genes. So all of this is called initiation. Then we get to the part that's called elongation. Elongation is where we're actually creating the RNA segment, the RNA messenger RNA, and we're doing that by moving down the DNA using the template strand, and we are matching, creating a complementary match in a long line of nucleotides here for RNA, for the messenger RNA. The last portion is termination. That's where we finish. So we have initiation, elongation, termination, and that's the spot where the RNA polymerase releases from the DNA. It goes back to being wound together in that double helix, and you have a finished, completed piece of RNA ready to move on to the next step. So as we mentioned, there's basically 3 stages to this. We have initiation, elongation, and then termination. I don't want you to get too bogged down in all of the details that we're about to discuss about initiation, elongation, and termination. I want you to know the basics, and I do want you to be familiar with perhaps some of the phrases or terms that you might hear later on in your college career, but I don't want you to get bogged down too much in the actual fine details of every little process here. In the picture we already looked at, we talked about the promoter. The promoter is the area where the RNA polymerase is going to attach to the DNA. That's not going to be part of the actual piece that's going to be transcribed. It's going to be a few dozen nucleotide pairs upstream from the starting point. And then we also have something called transcription factors. Transcription factors are just additional enzymes and proteins that kind of help out with this whole transcription process. And if you combine the RNA polymerase with all the various other transcription factors, they kind of build a enzyme machine, basically a combination of multiple proteins and enzymes. All of that together we call the transcription initiation complex. There is a funny little phrase that you might have heard before when you're talking about transcription called the ta-ta box. So I wanted to mention what the ta-ta box is because it kind of stands out as a funny phrase, and you may have heard it before. There's a specific promoter called the ta-ta box that's pretty important or crucial to signaling to the RNA polymerase where to begin transcription. So why would they call this particular area of DNA, this promoter, the ta-ta box? Well, if you look at this set of bases in the eukaryotic DNA sequence, it's T-A-T-A-A-A-T-A. Of course, that doesn't make any particular word in English, but it kind of looks like ta-ta. This particular sequence of nucleotide bases within the DNA is kind of a promoter region that signals, like I said, to the RNA polymerase, okay, this is where you need to latch on and begin the process. And that ta-ta box will not be part of what gets coded into the messenger RNA. Remember, it's going to be a little bit ahead or upstream from the actual transcription site, but it's kind of like the signal, the starting line for the whole process. So if you ever hear that funny phrase, ta-ta box, you know it is basically a promoter region in eukaryotic DNA for transcription. Of course, a picture is worth 1,001 words, so here we have an illustration. Here is that area called the ta-ta box. It's basically the beginning of the promoter region. This is the area where the RNA polymerase and all the various other enzymes are going to attach to begin the transcription process. So in this case, the top part is the non-template strand, and the bottom part is going to be what we're actually making a copy off of, or using that as the template. So here our transcription factors, which are some of those other proteins and enzymes, come into place, and then the RNA polymerase attaches there as well, unwinds the DNA, and begins the transcription process. So after initiation, we can move into what we call elongation of the RNA strand. As the RNA polymerase slowly moves down the DNA, it untwists that double helix, 10 to 20 bases at a time, and as it passes by, the double helix will kind of twist back together again. The transcription progresses at a rate, it says, of about 40 nucleotides per second in eukaryotes. And a gene can be transcribed simultaneously by several RNA polymerases. So what that means is you don't have to have just on a single strand of DNA one RNA polymerase working on it at a time. You could have 3 or 4 RNA polymerases kind of lined up behind each other like semi-trucks in a convoy, and they can all be moving down that DNA strand using it as a template to create the messenger RNA. The nucleotides are added to the 3' end of the growing RNA molecule, and once again, a picture always helps. So here we have a section of the double helix DNA. Notice it's unwound right here where we're working. This giant blob here is the RNA polymerase, and it is using this section of the DNA as its template, and it's taking the complementary base pairs, the RNA nucleotides, and putting them into place, creating a long chain in red here of the newly made RNA. You'll notice it's base paired just like the DNA, except the only difference is where in the case of DNA it's A and T. Well, the RNA doesn't have T, it has uracil instead. So instead of thymine, it has uracil. So where there is an A on the DNA, it won't match it to a T, it will match it to a U. So that's one of the few differences between the RNA and the DNA. The RNA is single-stranded, and it uses the U nucleotide base as opposed to the T nucleotide base. Finally, after initiation and elongation, we get to the end of the process, termination. How does the RNA polymerase know when it's done, when it's finished making its little section of messenger RNA? Well, the mechanisms are slightly different in bacteria, prokaryotic creatures versus eukaryotes. In bacteria, there is a section of the DNA that basically acts as a stop code and a start code. And so when it reaches that start code, the promoter region, it knows to begin, and when it reaches a specific stop code, it knows to stop. And at that point, the RNA is finished, and it can be floated off to the various protein manufacturers, the ribosomes, to get the whole protein manufacturing process underway. In eukaryotes, it's slightly different. The RNA polymerase does reach kind of a termination signal, but then it releases its messenger RNA piece. It uses something called polyadenylation, a polyadenylation signal as its stop piece, but the messenger RNA that gets released is not really clean and ready to go. Much like we kind of mentioned previously, this initial RNA transcription is kind of like your boss hastily writing directions out to give up to the whole company. Well, those directions are actually probably going to go to his executive assistant or secretary, and he or she is going to clean them up and proofread them and spell check them before sending them out to the entire company. Well, that's how it works in eukaryotes. The initial RNA that comes out of this process still needs to have some work done to it. It needs to be cleaned up, and we'll talk about that more a little bit later. All right, so why don't we take a break right there? That's the transcription process. That includes initiation, elongation, and then termination. And then we'll get into what happens after that initial RNA is made. What do the eukaryotic cells do to it before they send it out to the ribosomes? That'll be our next part. Take a break. Come back in just a moment.